Hello, everyone. Thank you for attending today's webinar, Transforming the DMTA Cycle Through Enhanced Collaboration, presented by Cresset. I'm Rebecca Kushmiter. I'd like to start by introducing today's speakers. We will be joined by Steve Jurich from Discovery Chemistry and Technology Consulting, LLC, Sabina Ruppel from Akana Oncology, Louis Vidler from Eli Lilly and Company, Lou Whitehead at Acelot, and Rob Scoffin at Cresset. You can read their full bios on the left side of your window by selecting the Speakers tab. Just a few notes before we begin. To access additional resources for today's presentation, please click the Handouts tab button on the left side of your screen. You can access closed captions from the bottom right corner of the video player. This webinar is being recorded. It will be available to watch on demand within 24 hours. We'd love to hear from you during the presentation. Please submit any questions you have using the Questions and Answers tab on the left side of your screen. We will conclude with a Q&A session, time permitting. Okay, let's begin. Rob, please go ahead.
Thank you, and thank you to FierceBiotech for hosting the seminar. I'll say a few words of opening introduction about myself and about Cresset. So I've been in the cheminformatics molecular modeling industry for 32 years currently, and have worked in a variety of roles within both software development and project management, and more latterly in senior management roles within several organizations. And for the last 13 years I've worked at Cresset, where I lead the team and lead the strategy in developing applications to help customers in drug discovery research. And primarily we focus on in silico solutions, so helping customers through a variety of applications and platforms, both detailed design work in 3D molecular modeling, in cheminformatics, and more recently in focusing around the process of research in DMTA. And so we have launched a platform called TRX, which focuses on the design, make, test, analyze cycle, DMTA, and provides seamless collaboration and communication to the sometimes very large teams that can get involved in a research process. The background to which is really sort of focusing around this, the challenges that we see in drug discovery. And one analogy that I use quite frequently is that of structural engineers or civil engineers who stopped building structures by constructing them and then seeing if they fell down about 200 years ago. And seemingly only in the last sort of 15 to 20 years as it's sort of become very commonplace in the pharmaceutical industry not to just make some molecules and see if they work. And so what we do as a company is to focus on trying to simulate and calculate as much about the molecules as we can before committing to a design. So effectively helping customers to get to an endpoint sooner by doing less chemistry and less biology. And the background to that is obviously drug discovery is a very complex process, very expensive process, takes years and billions of dollars to get a successful product to market. And the costs of that R&D process are ever increasing, largely driven not just by inflation in underlying costs, but also in ever increasing sort of complexity around the approval process and regulatory hurdles. And so one of the focuses is to try and be as knowledgeable as early as possible in that cycle of process. So using the in silico tools to inform at every step to make better decisions, to get better molecules. So with that, I will move to the individual panel members. So I'll thank them collectively before I make individual introductions. And I'm going to ask each of them to say a few words about themselves before we get into the specific questions. So our first speaker is Steve Jurich. And Steve, if you'd like to say something about yourself.
Sure. Yeah. Thank you, Rob. Yeah, I think Rob and I first met when I was global head of medicinal chemistry and technology at Abbott, which then became AbbVie. Um, we had a number of successful collaborations. Then my responsibilities there, apart from medicinal chemistry, in the technology organization, we were responsible for a variety of disciplines: high throughput chemistry, high throughput experimentation, chemical biology, chemical proteomics, targeted protein degradation, and also we had the structure based drug design organization within our bailiwick as well, which included additional CAD, protein crystallization, and so on and so forth. So it was in summary, it was a large smorgasbord of different technologies which were brought together to support the global AbbVie medicinal chemistry organization, which was quite an effort because at that point I think AbbVie was probably second, third largest pharmaceutical company there. So after my time at AbbVie, this is a couple of years ago, I retired and started consulting. And I've worked since then. I'm on a scientific journal editorial boards, but I consult with a number of major players and also biotech or startup companies. So one nice thing about that is I've grown to understand a very broad perspective on needs of obviously major pharma, which I was pretty familiar with, but in terms of needs of smaller biotech startup companies as well. And I hope to be able to use some of those learnings to help with the discussion today. So I think I'll cut it off there, Rob, and let you move on.
Thanks very much, Steve. And next we have Sabina Rupel from iCana Oncology. Sabina, if you'd like to say something about yourself.
Thanks, Rob. So I currently hold the role of VP of Drug Discovery at iCana Oncology, where I lead a multidisciplinary team that is responsible for everything from early discovery all the way to DC nomination. Prior to that, I was head of medicinal chemistry at iCana. I'm a trained computational chemist, and that's my connection actually to Cresset, which goes way back to Boehringer Ingelheim, where I first started working with Cresset tools. We had the whole tool chest back then, and currently we also have some products of Cresset at iCana. I had previous roles in various big and small biotech companies during my career as a computational chemist. And then more recently, I took on project chemistry leadership roles and delivered a BRD9 degrader that is currently in phase 1 clinical trials.
Thanks very much, Sabina. And if I'll move on to Lewis Vidler. Lewis, if you'd like to introduce yourself.
Yeah, hi everyone. Lewis Vidler. I'm a computational medicinal chemist, and I kind of operate sort of between those two disciplines. Many years ago, trained as a synthetic organic chemist, and then a sort of PhD level transitioned towards computational chemistry. That was at the Institute of Cancer Research in South London, kind of leaders in academic drug discovery. Out of that, I joined Lilly. That was about 10 years ago now. And I was at Lilly until 2020 when they shut the research site in the UK. I joined UCB after that, so went from kind of big pharma to medium pharma, continuing to work on projects, which is pretty much what I've done throughout my career in many therapeutic areas. I was with UCB for about 18 months, and then I joined Amphista Therapeutics. So from big pharma to medium pharma, then to kind of small biotech, as kind of the first computational chemist at Amphista Therapeutics. And we kind of went down a fairly Cresset heavy route. We licensed both Flare and the kind of Talks platform. And then in February this year, I rejoined Lilly, still in the UK. And yeah, I've been there for the last, 7, nearly 8 months. So yeah, thank you.
Very good. Thanks, Lewis. And finally, the other Lewis, who we're going to refer to as Lou today to avoid confusion. So, Lou, if you'd like to introduce yourself.
Yeah, good morning, good afternoon, good evening from Oyster Point in South San Francisco. My name is Lou. I am VP of Research at Acelot. Acelot is a seed stage biotech company focusing on neurological diseases. It's a spin out of the University of California, Santa Barbara. I trained as a chemist at the University of St. Andrews. I have a PhD in computational biophysics from the University of Southampton. Did an industrial postdoc at Novartis from 1999 to 2002. Continued at Novartis in the United States. The move from New Jersey to Cambridge and Tech Square in 2002 and 2003 in the NIBR organization. Structure-based design, ligand-based design as a computational chemist that evolved and matured into a computational medicinal chemist. I left Novartis to go to Nimbus as their first non-Schrodinger computational chemist as a strategic pivot that that organization was going under as those two organizations kind of were understanding which way they were going. I had a wonderful time there, moved to a Series A startup in Boston for a couple of years and moved to Acelot earlier this year. I do still live in Boston. My commute to San Francisco is intercontinental. I do this every couple of months, and I think that kind of reflects the kind of globalization of how all our skills on the panel are helping drug discovery for the patients that are waiting for treatments.
Brilliant. Thanks, Lou. And thanks to all for agreeing to be part of the panel discussion. So we split the discussion up into broadly 3 areas, looking at the changes that are happening within drug discovery, looking at potential solutions to some of those challenges and changes, and then looking at one specific change that's clearly sort of has the potential to be disruptive in the industry around AI. And so we'll take those in order. And for the audience, we're taking this question, drug discovery, what has changed in recent years and what longer term trends have been accelerated by the COVID-19 pandemic. As part of each of the discussion groups, we're going to put a poll up on screen to get your participation. So we'll have questions and they'll appear on screen like this. And so the first poll, the question that we're asking is, what do you consider to be the biggest challenge caused by changes in working patterns within drug discovery over recent years? And the options that we provided you with, and we'd appreciate if you would make your choice whilst I read them out. So number 1, managing relationships associated with the increasing number of partnerships with external vendors, including CROs, biotechs, etc. Number 2 is the straightforward remote working. Number 3 is the move towards cloud-based computing. And number 4 is difficulties in recruiting for key roles, a challenge we've all probably faced over the last few years. So that was, what do you consider to be the biggest challenge caused by changes in working patterns within drug discovery over recent years? Number 1 was managing relationships associated with the increasing number of partnerships with external vendors, including CROs and biotechs. Number 2, remote working. Number 3, the move towards cloud-based computing. And number 4, difficulties in recruiting for key roles. So we're going to use the poll results as a context for some of the discussion on the key topics. I'll give the panel members a chance to sort of look at the results that have come in. But broadly speaking, we've kind of got a 50-50 split there between managing relationships and recruitment. Nobody seems to be phased at all by the move towards cloud-based computing, which is great to hear. And then a smaller challenge around the sort of move to remote working. So in terms of the question that we posed in terms of the challenges, I thought it'd be good to get each of the panel members to use their experience to talk about some of the challenges that they've seen. And if I'll take the opportunity, Sabina, to ask you to start in terms of what you've seen. You described a career that's gone from sort of major pharmaceutical company into a series of biotechs, and so obviously you've got a good background in terms of seeing different organizations and different organizational structures, and some of the sort of challenges that might arise in that respect.
I think one of my main challenges currently as a team lead is really recruitment. That is extremely difficult and competitive in the Boston area where ICANNA is located. And so we actually end up working with a large number of consultants routinely all over the world in different time zones. And so that poses the challenge that you have to have some sort of communication tools that are accessible really all around the globe in order to ensure that the information flows seamlessly between all the parties that are involved, and no delays result just from emails not being read or simple things falling by the wayside due to non-modern technologies. So for us, that's a main concern as a very small biotech.
And could I, Steve, sort of, I think you described also a sort of a parallel journey of going from one of the biggest of the pharmaceutical companies into now a role where you're interfacing to multiple companies. Would you sort of reflect on some of the challenges that you've seen in that process?
Well, yes, reflecting on Sabine's comments, I mean, recruiting is very different at a major pharma level versus startup/biotech. And I think for biotech, it's it can be difficult because I've discussed this with a number of friends. You know better than I do. I mean, recruiting quality CAD people is difficult, remains difficult. What's your source of those? As a limited supply, right? So I think that's pretty clear. I know that you've been very focused on that over the years. In a more chemistry-centric explosion of startups, there is a limited number of qualified chemists to become, say, VP of a startup company. And so that's a challenge to recruit quality people at a good level. And I've seen companies that have done very well in that, but also ones that have struggled to recruit really quality people. And so there's a couple of my clients, they've been looking for VPs for MedChem or MedChem Discovery for a year, can't really get good candidates because of the smaller pool, and there's a lot of openings within biotech. Plus, MedChem's changed. I mean, a lot of people now are trying well, I mean, a lot is an ambiguous term, but there's a lot more chemical biology out there, and so a different degree of training. Synthesis training is somewhat different. There's been a change in focus on that. So the phenotype of people that you can now recruit has been changing. So this remains a current challenge in my experience.
Yeah, I think we all see those challenges in those sort of different verticals. Lewis, in terms of the kind of communication challenges, what specifically have you seen as you've joined an organisation where the UK Research Centre was closed, you've rejoined that organisation sort of back, still sort of now, is that are there different challenges that arise now that you've rejoined the organisation and working somewhat remotely?
Yeah, for sure. I mean, if you're going to work remotely, you always miss out on those kind of corridor conversations and kind of replacing, especially when you've got, if you're all remote, you're all in the same boat. Whereas if you're working remotely with a group that are frequently together, there's definitely challenges there. I mean, what's interesting returning to an organization that I was at previously is actually lots of those relationships I had before, I still have. And for me, building those relationships in person, that's kind of the key. So it's okay to be remote, but being able to catch up with folks in person, being able to travel, and actually it's not just the work, right? It's being able to go to lunch together. It's being able to go out after work as well. That really helps. So actually rejoining Lilly has been kind of interesting because I know lots of people. Contrasting that, when I joined UCB during lockdown. And when I joined, they were just starting to pilot people coming back to work. And then after a short while, people were coming back and then they locked down again. And so I never really got to build those relationships with the people outside of just working with them on projects. And what ended up happening was I was working with actually more of the folks in Belgium. UCB are kind of split UK and Belgium. I was working on projects more with the people in Belgium and actually built more relationships with them. And so actually my decision to leave was actually far easier because I didn't have so many ties to the people. And then when I joined Amphista, I was also remote. But the organization was much smaller. So I was able to then put time in the calendar. I could have one-to-ones with kind of everyone within chemistry, and it was not taking up my entire week doing that. So I think the move to remote working, I really appreciate it, and I like having the flexibility, but I think you need to be proactive with building those relationships. If you're very complacent, that's actually when things really struggle, and then you start having miscommunications and all those things that come with lack of relationship.
Is Lou, is that something that you have seen, particularly now in your intercontinental commute, transcontinental commute?
Yeah, I mean, absolutely. I was just to Lewis's comments. I think the benefit of being remote as well, certainly as our chemistry is done in India. We also have other relationships that we manage in Western Europe. Being situated in Massachusetts, I'm in a kind of sweet spot zone where I can kind of speak to India at the start of the day when my colleagues in San Francisco, haven't woken up yet. But there's certainly the software tools that are out there are helping me a great deal. I do resonate with that lack of corridor conversations. Some of the best achievements in my career have been corridor conversations of things that I just wasn't expecting, came out of the blue, and I ran with them. I can't obviously quantify that, not having them, but I come here at the end of every quarter, so I kind of make up for it that way. So thank goodness for Zoom and Teams and Talks. It certainly fills those gaps that allows me to do my remote working just as optimally as if I was in the office.
Yeah, I often comment to people that it's incredibly hard to schedule creativity. So pushing people into a room and shouting "be creative" at them appears not to work.
Yeah.
So in terms of, I think that sort of set the scene really nicely, and thank you everybody for those sort of reflections from your experience. we teed up now the kind of the next step, which is to think about potential solutions to the problem. So if we look at question 2, we phrase that as how can technology help to address these challenges by improving collaboration, so improving communication and collaboration as we've identified those specific challenges. We do have a poll question for that as well. So the poll question audience, wake the audience up and get you to push some buttons. Number 2 is, what is the most important consideration for your organization when evaluating a new technology solution? And the options there are ease of use or ease of integration, good data security, ease of data sharing, and scalability of the solution. So that was, what is the most important consideration for your organization when evaluating a new technology solution? And the options are ease of use, ease of integration, good data security, ease of data sharing, and scalability of the solution. And we'll take a look at the instant poll results. So actually, more of a distribution and a dominance there with ease of use, ease of integration. We didn't differentiate those two, so it's not clear whether it was ease of use or ease of integration that's driven that sort of response. But then the data security clearly important, and then data sharing, and then scalability. I guess that's a challenge that as IT professionals we worry about, but less so as users. So in terms of the technology solutions, I think it's interesting, given the challenges that we've identified, and that both we thought about we heard about the recruitment challenges, but also heard about the post-recruitment integration into new organizations. Lewis, you made comments about the sort of the stickiness of your sort of interface to a new organization if you haven't built those networks person to person. And we also heard about the sort of challenges of creativity and communication and missing out on those kind of water cooler, coffee machine, corridor conversations. So in terms of sort of the applications of technology that you've seen in your careers to trying to address those. So Lewis, are there particular things that you've seen or used or had experience of that have helped?
I mean, the first thing I would say is actually technology just to allow people to have the time to then engage with each other. So like removing kind of, what you might describe as like handle turning, right? And computationally, that's things like, file manipulations and stuff, like everyone running the same database query and then doing the same steps after that. And, automation as a place to, minimize just where people aren't able to be creative, aren't able to communicate. I think there's a piece there. The other thing then is around kind of communication. We have platforms like this where we can talk on calls, but then also sharing of kind of scientific information, right? So when I was at Amphista, we were a small organization, but we leveraged a lot of CRO work, and so talks there really facilitate that. In terms of the sort of feedback and what people liked about the tool, we also had the sort of design side, but the make side, just managing the relationship with the CROs was hugely impactful there. And actually it's linked to that taking away of just the handle turning, right? Automating work, making slide decks or getting slide decks from the CRO, then they're out of date, chasing them. When you hire smart scientists and you pay them good money for doing that, you want to maximize their time being creative and doing science, not doing administrative work and file manipulations. And so technology has a huge role in, I see it the exact same way we use machines to automate our physical work. We use a computer to automate our, thinking tasks, or the basic thinking tasks. And I see that only going in one direction.
Do you, if I sort of address this to Lou, do you see that in a similar way from your experience and you in having worked through sort of both big organizations and small organizations, those same kind of applications of technology to remove the drudgery? Does that have an impact versus applications of technology to address sort of hard to process challenges? I guess it would be what as computational chemists we kind of look at hard problems a lot of the time, and actually it was interesting what Lewis was saying there was sort of take away some of the drudgery and it opens up the space for you to think about the hard problems.
Yeah, I think as I reflect, certainly to my time at Novartis, that was an organization that in the mid-90s, I don't know whether it was by accident or by design, but their cheminformatics platforms were probably a decade ahead of everybody else. So I remember beginning in that organization, and the platform that was being used at the time to access information and chemical information. Certainly 2 or 3 years after a large merger, Ciba and Sandoz had merged a couple of years before, so they'd gone through that kind of headache. Having access to the data incredibly quickly was, in my opinion, was a huge competitive advantage. Obviously, as time went on during my time there, those infrastructural software tools needed to be updated. Certainly the move to the web was in the early around 2010, 2011, could have gone a little smoother. I don't know whether that reflects the status of the technology at that time. I did feel like we were taking a couple of steps back from what we had before, but I wasn't directly involved in that. But the ease of use of cheminformatics, I think, really benefited. Later on, the harder problems, I would say certainly the rather amazing increase in high performance computing and cloud, I think kind of broke the doors off what computational chemistry could do, whether you're looking at analyzing data, thinking about running FEP, if you want to go in that direction, screening very ultra large libraries. So from my perspective, I think I've benefited from 2 eras in multiple organizations that have done some things incredibly well. And as I've moved to smaller companies, it has been quite insightful the way that my brain works in terms of what are the things that I'm going to pick first to build, because I know this is our immediate goal, and this is the easiest way to achieve that goal with the minimum amount of effort, because you have to be a bit scrappy.
And I was wondering, Sabina, if you had sort of similar experience, and again, given a experience in organisations both large and small, have you found different sort of technology implementations have been sort of useful or not useful, and any learnings that you might like to share around that?
Yeah, I think in big organizations, at least in my experience, you often have internal tools available, internal technology platforms that are built in collaboration with IT teams, for example. And when I was at Boehringer Ingelheim, for example, we had platforms like that were really customized to the workflows as they were happening at BI, and the IT support was always there to update things, to make it work flawlessly, truly. And it was adopted throughout the medicinal chemistry group, which was a fairly large group, over 100 people at one point in time. And so I found that very useful. And moving to small biotech, I realized that these solutions are not common at all. And I had, in my first small biotech, we had somebody who was very interested in automating workflows and therefore implemented a commercial solution at that time. It was Life Design, and that was already extremely helpful for collaborative efforts within the organization. But I have to say, we had in another small biotech I worked at, we did build an internal solution, and again, it was customized to our particular workflows, and that's always great. And you have the person there when you need something; they will do exactly what you need to get you to do most expeditiously what needs to be done. But when this person leaves, you're left with essentially nothing, because nobody else is willing or able to take on changing these tools. And so when I took over medicinal chemistry and now drug discovery at ICANNA, for me, the most important goal was to have a commercial solution that's easily implementable, because I don't want to burden a very small IT team even more with things that is maybe not their top priority either. So ease of integration with any existing solution was really on top of my list, as well as vendor support for anything that goes wrong, because internally we just don't have the capabilities to do anything in terms of what I call research IT. And then also, truly for me, it was important that it can be accessed across the globe. So by our CROs, not just one, but potentially multiple CROs, chemistry CROs across the globe, as well as a number of different consultants. So I have consultants in the UK, I have other consultants in the US, and it's really hugely helpful to have a platform, and our current platform is Torx, to bring together all these ideas, capture the ideas, but then also for my one medicinal chemist as a project lead to be able to easily follow the work that our 40 CRO chemists on a project do across the globe, because it's still a lot of information for one person to handle at any given time, right? So the solution really has to satisfy many different things, like enabling the one project lead to be on top of the data all the time, every broad accessibility across the globe, ease of integration, and really strong vendor support. That for me was the deciding factor when I choose these collaborative solutions.
Great, thank you. And Steve, I was going to actually direct a specific question at you. And the specific question was, given your experience in working within Abbott, then AbbVie, with very large chemistry teams, but deploying innovative solutions, including deploying sort of, if you like, complex computational chemistry applications, but within that kind of medicinal chemistry environment. And in the context of our poll question around ease of use, what was your experience in terms of the success or otherwise of trying to sort of bring those two worlds, that computational chemistry world and that sort of medicinal chemistry world, together around a platform?
Okay, so this is an interesting question. So, as you are aware, I mean, AbbVie, we had, for technology, we had a multi-million-dollar budget every year for technology acquisitions, right? So you had to look at, for chemistry, what can you bring to the table, right? So two things, is reduction in cycle time and reduction of cost. Right? There's two things that chemistry brings to the table. Also, it's important, I think, to remember a comment from George Whitesides from Lab on a Chip. If you develop new technology, if it's any use, people will buy it. Okay. And as a provider for our technology, we were providing global support in the technology area at AbbVie, which was, as you're aware, a large organization. What you develop has to be used, right? Otherwise, as a VP, you will have a short half-life because you're providing stuff that has little value, right? So, and there is a risk at times of over-engineering solutions because you have a lot of money and it's like, wow, this would be fantastic to do. And, for example, I think we were going to talk about, the design, make, test, analyze cycle. I mean, we developed very nice solutions to that, which sort of fit. But I think, Lou would comment at some point on, say, the Lilly Life Science Studio, which was a huge engineering feat. Did that provide value? So, those are all important, right? You also face the challenge of it is relatively easy to come up with new ideas. It is often extremely difficult to get rid of the old ones because within an organization, right, you have people who are like, well, this is the way we did it. This is the way we're used to doing it. Changing the culture can be a big problem. So you have to have solutions, whether it's chemistry, fully integrated closed loop stuff that Brian Warrington at GSK was trying to do years ago, and integrate chem informatics tools into that. It's great if they work and they are reliable, right? And as you well know, if you bring in new software tools, and and they suggest perhaps helps you filter ideas and make selections, and you make a molecule or a series of molecules that is 10, 15 steps, and these things are inactive, your CAD support person will become your worst enemy instead of your best friend. So it is, the key thing is the introduction of robust technologies that have broad uptake within an organization. I think that's absolutely clear based on my experience. Now, if you're looking for stuff on the, and I'm happy to talk more about the specific applications to the design, make, test cycle. So I hope you're going to ask me on that one later on.
But we'll come to that in the Q&A. And sorry,
outside for technology
sorry, just in the interest of time, I think we need to move on to question 3, and then I'll loop back because we've got time in the Q&A.
Okay.
So we'll loop back. Apologies. So actually, in terms of the third question, and I think this is a really nice segue from what Steve was saying in terms of introduction of novel technologies and the need for those to be reliable and robust. So slightly provocatively, we wanted to sort of address the question around artificial intelligence, machine learning, which has seen a massive increase in application over the last few years, both in all spheres. So we're seeing sort of AI tools like ChatGPT-4 being deployed everywhere, and so we've all sort of seen that in the popular media. But there are many applications within life sciences discovery, drug discovery. We wanted to just spend a little bit of time sort of talking about that. We have a poll question for that, and the poll question is, where in the hype curve do you think AI technologies are with respect to drug discovery? And the options we're provided there are too early to tell, overhyped, in the valley of disappointment, or accepted as a commonplace tool. So where in the hype curve do you think AI technologies are with respect to drug discovery? And it's too early to tell, overhyped, in the valley of disappointment, or accepted as a commonplace tool. I'll give the audience a couple more seconds to click on the answers there. And drum roll. So we've got 50% roughly in the overhyped bucket, which is, I think that's going to fire up the conversation, and then a good number in the too early to tell, and a few lucky souls who work in the world of AI-driven drug discovery who accept it as a commonplace tool. So in terms of the sort of deployment of AI, how much of that, and apologies everyone, we this is going to have to be fairly rapid fire. So in a minute or less, what applications have you seen, Steve, that really makes sense for AI today? So where would you point at it and say, actually, it's in the commonplace tool rather than it being overhyped or too early to tell?
It's used routinely. I think probably so I should say, firstly, I'm on the board of 3 AI companies, and I think they would probably agree there's some degree of overhype. Targets that have been worked on have been sort of more well-established one than some of the novel targets that we're currently working on. So value is, I think, data analysis, not just in a med-chem area, but it could be for clinical trial selection, off-target assessment of drugs, large data analysis of difficult problems, have all cell painting analysis, whatever. And I think that's going to have tremendous value. It's also going to have tremendous value for med-chem in the future. It's getting there, not there yet.
Lou, how about you?
I mean, I've seen generative AI in terms of chemical space that a medicinal chemist or a computational chemist would give a thumbs down of, don't bother making that compound. It will not be active on that target. I'm balanced enough to let that type of compound go. I've seen those compounds get made and become hits. And to me, that was what is going on here? I do align with the hype cycle a little bit, but I do feel like I've been doing AI and ML for a couple of decades. It's just been rebranded. But I do think that there's some excitement in the generative chemistry space that kind of takes the burden off the eyes of a medicinal chemist and a chemist to put in a little bit more risk.
And Sabina.
Yeah, I think in my organization, people constantly ask, can we do something AI? Can we deploy AI? And it is pretty much across any kind of discipline or project. And I think what I find is that there needs to be a lot of education happening with the C-suite and potentially board members, because I think there's often a misunderstanding as to what problems are amenable to potential AI solutions, and that it's not something that you can just throw at every problem you encounter. And I think that's something that we as computational chemists or scientists are able to potentially help organizations to better understand where these solutions could actually have an impact, rather than randomly deploying it across the board and then only to find out, oh, this actually didn't work. Let's not do this again. So I think there's a big responsibility from our side to really educating people and deploying it the right way.
Great, thank you. And finally, Lewis.
Yeah, I mean, I agree with Lou to a certain extent. I mean, the field of computational chemistry is 30 plus years old, right? And if we look at kind of progress within that, it's probably kind of linear, maybe lightly exponential. However, kind of expectation within that is all over the place, right? The in the very early days is, we're going to make every molecule, you medicinal chemists are going to be redundant, and then suddenly you're into that kind of trough of disillusionment, and there's not much interest. In the current space, is like, we can tell the difference between a cat and a croissant using an algorithm. And therefore we could design drugs using a computer. So, because there's been progresses in other spaces, and image recognition actually being a really big one, suddenly there's an expectation that same progress is going to be made elsewhere. And so at the moment, I think it's overhyped because of the size of the expectation. And there's no doubt good science going on in the areas of generative design, in the areas of deep learning models and things like that. But the expectation is far more than realistically what they can give. And so actually you're going to end up disappointed, or you as in I guess in you globally, people are going to end up disappointed because actually the delivery won't match that expectation. But then those technologies will just become standard practice, as pretty much every technology has.
Yeah. I genuinely had somebody ask me the question, who's an AI scientist working in an AI drug discovery company, how come the medicinal chemists were refusing to make all the molecules that he'd designed on his computer? And I think he genuinely felt that they could have made them if they'd wanted to, they just were refusing to. as opposed to, you've designed me something that just blows up, and I'm not making that. Thank you. That was great, and that was very rapid fire. Apologies for moving that through relatively quickly. So we have left time for Q&A on the topics that have been raised. We've got some questions coming in, and one of the questions that's come in is actually addressed to Lou, and the question is, could you provide an example where you've seen the collaboration tool making a game-changing decision?
Yeah, I mean, it goes back to the start of my career as an industrial postdoc. At 99, 2000, Accelris had a product called DS Viewer, and they rolled out an upgrade where they put an ActiveX controller, so you could put your computational models, X-ray crystal structures on the web. And I was, relatively green computational medicinal chemist, but one thing that I did see as an incredible value is the work that I'm doing on, essentially it was a PI3 kinase project. I was the first chemist assigned in the Novartis programs. The work that I'm doing, if I can share it with my chemistry team, they can understand what I'm trying to persuade them to make, and maybe they can do their own independent work. And it was kind of being able to go to my colleagues' offices and say, type in this web page and the model will show up. We can interact with the model and we can design it. I mean, I worked with incredibly talented group of chemists, but Catherine LeBlanc, who is still with that organization in Switzerland, who came to my desk one day and asked to do a specific urea library substitution. That was performed primarily on what you could see from the models. That SAR that resulted from that conversation and that platform is within PICRAE, which was FDA approved 3 or 4 years ago. So, the designs that I was involved in 2000, 2001, the SAR evolved, but the chemical matter within that compound, I think that bought us a year or two, and I might be being, we might have missed that, and so I've always, since that point, been very conscious of that kind of sharing and collaboration. I'm not the world's expert in anything, but that for me was a total game changer.
Brilliant. Thanks, Lou. We've got another question in, actually, which is, I guess the flip side of what we've been talking about in terms of communication and sharing, which is the security question. So actually addressed to Lewis, and the question was: is there more emphasis on security in larger organisations rather than smaller organisations? But I guess we've got 5 minutes left, so everybody could chip in on that one.
I mean, I would say everywhere I've been at, security is a big concern, and actually in smaller organizations, often, big pharma is a place where a lot of people train. So the people who end up in smaller organizations probably have come from a big pharma background, and most big pharmas drill into you that security is very important. So I think regardless, I think it's just a requirement. I just see it as kind of standard practice that, when you're working with chemical intellectual property, you make sure that it's safe and secure, because that's where the value of the organization comes from. And without it, there almost is no organization.
Sabina or Steve, do you want to add anything to that?
Well, I can just say briefly, big pharma security is a huge concern. There was certainly, as an example, at AbbVie and one at Merck, where the site was down because of malware downloaded externally into the systems. So it was a huge deal. The other big thing is some people, small companies in particular, are concerned about security of data with working with CROs predominantly in China. And I don't personally think that that's an issue at all. I mean, there's one bad example of that, which people probably are aware of, but that was a rarity, and we never mostly major pharma don't have a problem because they do loads of outsourcing with China. So I'll leave it at that.
Yeah, I second what Louis said. I've worked in big and small pharma, and I think data security is a huge concern regardless of the organizational size. I think what makes it potentially more challenging for a smaller organization, if you have tools, for example, like Torx, that internally you don't have a lot of support helping with the implementation, and you have to make sure that your IT team, which often is just one person, right, understands what the tool can bring in terms of efficiencies and return on investment, essentially, by sacrificing extra time to find solutions that fulfill the data security requirements of an organization. So I think that's more the challenge I see for a small organization. Whereas when you're very big, a lot of it is just done internally in the first place. So your data never leaves your organization, especially if you have these internal solutions like we had at Boehringer Ingelheim, for example.
Thank you. And thank you all for a really wide-ranging discussion. Thank you for your engagement. And I will just wrap up with a brief sort of summary of the discussion that we've heard. I think the topics that came through strongly there were certainly the strength that comes from being able to collaborate and share information and thoughts with colleagues, not just next door, but across the globe. I think we heard quite strongly the need to build relationships and the challenges around building relationships when organizations are remote or when you're working with partners. And so the collaboration tools become a very good platform to enable that. And we also heard about some of the challenges of deploying new technologies. I think the robust word was used, and I think that's a particular challenge. And we used AI as an exemplar of new technologies which are being deployed and which are being talked about. And from an audience perspective are being possibly overhyped. But as they become more reliable and robust and trusted, they become everyday tools. And I think, as Lewis and Lou both said, we've been doing AI ML for 30 years. It's just had a new badge, and that applies to lots of technologies that have been deployed across the drug discovery process over the years. So it just remains for me to say thank you very much to FierceBiotech for hosting the webinar. Thank you again to the speakers, the panel speakers, for fantastic contributions, for their thoughtfulness, and thank you for attending and for your questions and your input into the polls. And thank you very much. And if you have any questions, our contact details are on the slide. Thank you.
Oh, thank you very much. And thank you all for attending today's FierceBiotech webinar, for submitting so many great questions. I'd like to thank our speakers again for participating, and Cresset for presenting today's webinar. A recorded version of this webinar will be available for you to access within 24 hours using the same audience link that was sent to you earlier. Thank you again for joining us, and we look forward to seeing you at future events.